Hi, thanks for joining me today to hear more about the PubMLST Pneumococcal Genome Library. The pneumococcus, or Streptococcus pneumoniae, is recognized as a leading cause of bacterial pneumonia, meningitis, and bacteremia worldwide. And over the past 20 years, we've learned a lot more about the epidemiology, evolution, and biology of the pneumococcus thanks to genome sequencing. In 2001, the first pneumococcal genome sequence was generated. And since then, we've scaled up to analyzing several thousand pneumococcal genomes in a single study. Over the past 20 years, we've also seen a shift in the culture of data sharing, where publishers and funding bodies now require authors to make their data publicly available. This is fantastic, and it's resulted in a massive increase in the genomes available from public repositories, such as the Sequence Read Archive, where there are currently over 84,000 pneumococcal records available. Now, this is a great resource, but it's important to recognize that actually these data aren't easily accessible to a large variety of researchers. This is because you need to have the infrastructure and expertise to be able to retrieve, process, and manage these data. And it's this problem that drove us to create the Pneumococcal Genome Library. So it's worth just defining a genome library before we start. And what we mean by this is it's simply a curated set of genomes and metadata that are publicly available via the PubMLST interface. In particular, the Pneumococcal Genome Library forms part of the Global Meningitis Genome Library. And for those of you who work on Group B strep, Haemophilus influenzae, or Neisseria meningitidis, you might be interested to look at those genome libraries, which are available at the link that I've included at the bottom of the slide. In the case of pneumococcus, we focus on published genomes and associated metadata. And a good question to ask is, what's the advantage of having this information available in a library? And the answer is to do with querying and retrieving data. So when we start with the querying, you can search the genome library based on different fields. So you might want to retrieve an entire data set associated with publication, or perhaps you'd like to create a custom query where you recover all isolates from the UK that correspond to serotype 3 and also have a genetic sequence that you're interested in. Once you've run your query, you can then download the metadata and genomes from the database to analyze elsewhere, or you can carry out additional analyses within the database. And I should just say right at the outset that the library is currently available, and I've included a link to this on this current slide, and it will appear on future slides as well. So when we started thinking about how best to develop the library, we decided on a systematic approach. So the first step is a literature search. Then we obtain genomic data and basic metadata, just to say that this step has actually highlighted some obstacles for us. And I'll be coming back to that in a later slide. It's often an iterative process and deceptively simple in the outline. Once we've got the data, we then create genome assemblies if necessary, and then we upload both the genomes and metadata to PubMLST. After that, we do a genome quality check and then update the pneumococcal genome library. And it's worth highlighting here that this doesn't have to be a one-shot process, and we have actually just repeated it recently to update the library. And so we think of the library as an evolving resource that we hope to continue to update over time. So over the next couple of slides, I'm going to break down the individual steps, starting with the literature search. So we've now carried out multiple PubMed searches, and we've covered articles published from January 2000 to July 2020. We've screened over 1,000 articles, and we exclude any articles that aren't original research papers. We exclude any studies that included unpublished data from public repositories. And then our main criteria is that to be included, a paper must include at least one whole genome sequence from a naturally occurring Streptococcus pneumoniae isolate. So based on those exclusion criteria, we were left with 217 eligible articles. As I said earlier, the next step is to then go into those articles and try to find the genomic data and the supporting metadata. And this is a point where I just want to highlight some issues that we ran into, because I think that this provides an opportunity for us to reflect on practices as a community and hopefully improve as we move forward. So the first step was to try to find accession numbers in each paper. And this was very interesting to note that 19% of our articles didn't include data, so they didn't share their data. And what I think is worth highlighting is that this is still occurring. So authors choosing not to share their data. Then when we looked at the papers where accession numbers were provided, we ran into some additional problems with 26% of the articles. And this is what we consider to be a data integrity issue, where the number of genomes that were described in the paper doesn't correspond with the number of accessions provided, or perhaps the accessions were invalid, incorrect, or suppressed within the databases. And then rarely, sometimes we found that supplementary data tables didn't exist, or they did exist, but didn't include accession numbers as indicated in the text. So again, this is a plea to everybody, not just in the pneumococcal community, but when you're preparing data for submission, please do consider sharing your genomic data and checking your supplementary files before submission. So not all bad news, thankfully. We did actually manage to retrieve a lot of data, and we prepared the first version of the genome library for release last year. It contained over 8,700 isolates from 92 papers. And as you can see from the map up there, a wide geographic distribution. We've got the earliest isolate was from 1916, and most of the data do come from the early 2000s onwards. It's also worth highlighting the diversity of the data set. There are over 90 serotypes represented and over 1,070 STs. So when we started working with the library and looking at the genomic data, we realized very quickly that actually the quality of the genomic data is quite variable, in spite of the fact that these are all published genomes. And we loosely categorize them either as low, variable, or high quality. When we say low quality, we're talking about sequence data that failed to produce an assembly at all, or sequencing data that was contaminated with non-pneumococcal DNA, or perhaps unusually large or small genomes. I'm not going to talk much about high quality data, except to say that we believe there is quite a lot of high quality data in the database. But just to take a moment to flag up the variable quality data. So in this large mixed bag, there will be genomes that have large numbers of contigs, where we perhaps struggled to recover core genes, or where there were atypical MLST profiles, which is often a red flag for us. And what we think is worth highlighting here is that it would be useful for database users to have an idea of the quality of the genomes, so that they can then decide what they would or would not like to analyze. And the variable quality is really important, because some analyses might be feasible on these data and others might not. And so while we've been thinking about this, it's really become clear to us that the community would benefit from greater transparency around genome quality. And that stretches all the way from authors providing clearer descriptions in their papers, to when we're producing the pneumococcal genome library, to making clear how we assess genome quality, and then providing some indication of that in the database. And this is our next sort of goal that we're working towards. And in the future, we hope to make that more accessible in the database. But what is coming next for the database in terms of the genome library is we're updating it to version 2.0. So we're currently preparing this update. We've assembled over 15,000 additional genomes. The data are already available via PubMLST, but at the moment we're also linking these data to individual articles and assessing the quality. So watch this space. We plan to release this shortly. To sum up before I end, just to recap what the Genome Library is. It's a public resource available via PubMLST, and it aggregates pneumococcal genomes and metadata from the published literature. We've said already that version 1 is live. You can access it today, and it contains over 8,000 genomes. And we are hoping to release version 2 very soon, but you can already access an additional 15,000 genomes via the PubMLST interface. The whole process has highlighted that we as a community need to aim to improve in terms of making our data available, ensuring the quality of our supplementary files, and also ensuring the quality of the genomic data that we produce. And I think this is an important opportunity for us to have more conversations as a community about what constitutes a high quality genome, and metrics that we could all be using. In terms of future work for the library, we will continue to add data, and we also plan to incorporate that QC information into the database in a variety of ways. So to end off today, I just want to acknowledge three groups of people. Firstly, my collaborators on the Genome Library, particularly Andy, James, and Keith, who've been very involved in the practical elements of getting the database set up and the library visible and up and running. And then special thanks to our funders, the Wellcome Trust and Meningitis Research Foundation. And lastly, I'd like to acknowledge the pneumococcal community, many of whom do make their data available. And this is sort of a rogues gallery of some of the key players who've contributed to the data stored in the first version of the Genome Library. And thank you very much for joining the talk today. If you have any questions, comments, or suggestions, our details are available via the PubMLST website. We'd love to hear from you. So thanks again.